In 2012, 6-year-old Emily Whitehead was dying from leukemia until an experimental therapy revolutionized cancer treatment overnight. Scientists reprogrammed her immune cells to become cancer-killing machines, and she's now cancer-free over a decade later. This CAR T-cell breakthrough has saved thousands of lives with blood cancers. And now we're making significant progress in using CAR Ts against glioblastoma. While challenges remain, there's growing optimism that these approaches may finally improve outcomes for patients with this devastating disease. Hi, I'm Daniel Chafamo, a second-year medical student at the Perelman School of Medicine. Today, I'll discuss how we are adapting CAR T-cell therapy for glioblastoma by addressing the unique biological barriers these tumors present. I'll highlight recent clinical successes and the innovative engineering approaches that are bringing us closer to effective CAR T therapies for glioblastoma. Glioblastoma is the most common and most aggressive primary brain tumor in adults. Defined as an IDH wild-type WHO grade 4 astrocytoma, this devastating disease carries a dismal prognosis with median survival of less than 15 months. What makes GBM particularly challenging is its near universal recurrence despite aggressive multimodal therapy. There is a strong unmet need in the recurrent setting for FDA-approved therapies that consistently improve survival. The current standard of care for newly diagnosed GBM follows the STUP protocol established nearly 2 decades ago. Treatment begins with maximal safe surgical resection, followed by radiation and chemotherapy. When the tumor inevitably recurs, options become limited to systemic therapies with minimal efficacy or consideration of resection or re-irradiation when feasible. Inclusion in clinical trials remains a priority in the recurrent setting as we desperately need new approaches that can break through this therapeutic ceiling. So what exactly is CAR T-cell therapy? This treatment involves genetically engineering T cells, either the patient's own or from allogenic sources, to express a synthetic receptor called a chimeric antigen receptor, or CAR. These synthetic receptors are designed to recognize specific tumor-associated antigens with high affinity, effectively reprogramming the T cells to target and eliminate cancer cells. As you can see, modern CARs have three main components: the extracellular domain that recognizes and binds to specific tumor antigens, a transmembrane domain that anchors the receptor to the T cell, and intracellular signaling domains that activate the T cell once binding occurs. When a CAR T cell encounters its target, this binding triggers a cascade of immune responses. The T cells activate, multiply, release cytokines, and directly kill the tumor cells. The success of CAR T-cell therapy is evident in the growing list of FDA-approved products. As you can see, these approved therapies target CD19 or BCMA, which are expressed on B or plasma cells. The remarkable efficacy of these therapies has transformed outcomes for patients with previously refractory leukemias, lymphomas, and multiple myeloma. In the context of glioblastoma, while there are no FDA-approved CAR therapies yet, recent clinical trials from groups at the University of Pennsylvania, City of Hope, and Harvard have yielded promising results in using CAR T-cell therapy for glioblastoma. Published back-to-back just last year, these studies demonstrated both safety profiles and encouraging signs of therapeutic effectiveness, generating significant excitement and hope. These promising results build on significant progress in understanding and addressing the unique challenges that solid tumors present compared to blood cancers. The three main challenges that CAR T-cell therapy must overcome in the setting of glioblastoma are: 1, poor trafficking and infiltration of tumor; 2, a hostile tumor microenvironment; and 3, tumor heterogeneity and antigen loss. Now let's take a look at each challenge in more detail and discuss the innovative approaches being developed to overcome them. The first major hurdle involves getting the CAR T cells to the tumor site. The blood-brain barrier represents a formidable obstacle for CAR T-cell therapy in GBM. This specialized structure features tight junctions between endothelial cells that restrict paracellular transport, along with efflux transporters that actively remove foreign molecules. Less than 1% of systemically administered CAR T cells successfully reach brain tumors, a stark contrast to the robust trafficking seen in blood cancers. One straightforward approach to overcoming trafficking limitations is locoregional delivery. In this approach, CAR T cells are administered directly into cerebral ventricles, for example, using an Ommaya reservoir, enabling them to access brain tumors without needing to cross the restrictive blood-brain barrier. This approach was one of the key innovations behind the aforementioned trials that demonstrated rapid tumor regression. Beyond efficacy, local delivery offers the advantage of maximizing CAR T cell concentration at the tumor site while minimizing systemic toxicities like cytokine release syndrome. While local delivery offers an immediate solution to trafficking challenges, engineering CAR T cells to better navigate toward tumors represents a more elegant approach. GBM secretes specific chemokines that can be exploited for targeting. For instance, modifying CAR T cells to express CXCR1 or CXCR2 enables them to follow IL-8 gradients into tumors. Jean and colleagues demonstrated complete tumor regression in GBM models using such CXCR1, CXCR2 modified CAR T cells. Similarly, CCR2B engineering enhances migration towards CCL2 secreting GBM cells. Clinical translation of these approaches is already underway, with trials evaluating chemokine receptor modified CAR T cells. This strategy essentially co-opts the tumor's own signaling mechanism to improve therapeutic targeting. The second major challenge involves the highly immunosuppressive tumor microenvironment. GBM creates its immunosuppressive microenvironment through multiple mechanisms. Inhibitory cytokines like TGF-beta and IL-10 actively dampen T cell function, while immune checkpoint molecules such as PD-1 and TIM-3 induce T cell exhaustion. Metabolic challenges further complicate matters. Hypoxia, acidic pH, and nutrient deprivation all impair T cell activity. Additionally, immunosuppressive cells, including regulatory T cells, myeloid-derived suppressor cells, and tumor-associated macrophages, infiltrate GBM tumors and inhibit CAR T function. TGF-beta deserves special attention as a particularly potent immunosuppressive factor. Highly elevated in the GBM microenvironment, TGF-beta directly inhibits T cell proliferation and cytotoxic function. It also induces regulatory T cells that further suppress immune responses and promote M2 macrophage polarization, contributing to broader immunosuppression. Clinically, TGF-beta signaling correlates with poor outcomes in GBM patients. Given its central role in GBM immune evasion, targeting TGF-beta mediated suppression represents a high priority strategy for enhancing CAR T efficacy. Several innovative approaches have been developed to render CAR T cells resistant to TGF-beta suppression. One strategy employs a dominant negative TGF-beta receptor, which acts as a decoy that prevents signaling through the endogenous receptor. CAR T cells expressing this construct maintain cytotoxicity and proliferation even in TGF-beta-rich environments. Another clever approach uses switch receptors that convert TGF-beta suppressive signal into an activating IL-2 or IL-7 signal. The Narayan trial demonstrated clinical impact with this approach, showing over 30% reduction in PSA levels using PSMA targeting TGF-beta insensitive CAR T cells in prostate cancer. Importantly, TGF-beta resistant CAR T cells maintain stemness and memory phenotypes in solid tumors, enhancing their long-term efficacy. The third major challenge for CAR T therapy in glioblastoma involves tumor heterogeneity and antigen escape. Even if CAR T cells successfully reach the tumor and maintain function within the microenvironment, they must still recognize their targets. GBM's notorious heterogeneity complicates this recognition, necessitating sophisticated targeting strategies. Single-cell analysis has revealed extensive intratumoral heterogeneity in glioblastoma at the molecular level. Target antigens like EGFRV3, IL-13 receptor alpha 2, and HER2 show mosaic expression patterns with spatial heterogeneity, creating distinct clones in different tumor regions. This baseline heterogeneity is further complicated by temporal evolution under therapeutic pressure, leading to antigen loss. O'Rourke and colleagues reported EGFRV3 loss following CAR T therapy in GBM patients, a clear example of treatment-driven antigen escape. This shifting landscape necessitates multi-antigen targeting strategies to achieve durable responses in GBM. One solution to this challenge is a bivalent CAR design, which involves engineering CAR T cells that recognize two different tumor-associated antigens rather than just one. This dual targeting approach functions as an OR gate logic, meaning that the CAR T cells become activated when they encounter either of the target antigens. This is crucial because it allows the therapy to remain effective even if cancer cells downregulate or lose expression of one target antigen. The Bagley trial mentioned earlier showed promising responses with a bivalent CAR T cell product that simultaneously targets EGFR and IL-13 receptor alpha 2. T-cell engaging antibody molecules represent another innovative approach to address heterogeneity. Here, CAR T cells are engineered to secrete bispecific T cell engagers, expanding their targeting capability beyond the CAR itself. These bispecific T cell engagers function as a bridge, with one binding domain attaching to a tumor-associated antigen and another binding to CD3 on T cells. The Troy trial mentioned earlier used this approach and showed rapid tumor regression. What makes this approach particularly powerful is the recruitment of endogenous T cells, which amplifies the anti-tumor response. This creates a second wave attack against tumor-associated antigens not directly targeted by the CAR. Finally, synthetic notch or SynNotch technology enables precision recognition with sophisticated if-then gate logic. Unlike tandem CARs that operate on OR logic, SynNotch requires the presence of two antigens for activation, reducing off-tumor toxicity in tissues that express only one antigen. The mechanism is elegant. The first antigen recognition triggers CAR expression for the second antigen, creating a sequential recognition process. Roybal and colleagues demonstrated selective killing of dual antigen positive cells while sparing single antigen cells. This approach is now in clinical translation with a phase 1 trial testing SynNotch CAR T cells in glioblastoma. Wrapping up, we've seen how early clinical successes demonstrate that strategic engineering and delivery approaches are transforming CAR T cell therapy for glioblastoma. The innovative CAR T approaches outlined today, from locoregional delivery to TGF-beta resistant designs to multi-antigen targeting, are showing unprecedented promise in early clinical trials. The field is now advancing beyond feasibility demonstrations towards durable efficacy. The integration of multiple innovations within single therapeutic platforms will likely be required to achieve transformative results seen in hematologic malignancies. Imagine CAR T cells that simultaneously bypass the blood-brain barrier, resist immunosuppression, and target heterogeneous tumor antigens. Such comprehensive approaches may finally unlock the full potential of cellular immunotherapy for GBM patients who have waited far too long for effective therapies. Thank you for your attention.